Good evening, everyone. Kia ora, malo lele, talofa lava, kia orana, malo ni, nisa bolavinaka, whakalofalahi atu, Māori, aloha. Warm Pacific greetings to you all. O ki ngoko Diana, me Aotearoa New Zealand, o ku tamai mo ku whae me Tonga, me hapai mo vavao. Tēnā koutou, tēnā koutou, tēnā koutou katoa. Welcome everyone to the University of Otago o Tako Whakaihu Waka on this very, very special evening and occasion. We are celebrating Professor John Ashton's Inaugural Professorial lecture together. And John, please know we are so very proud of you. My name is Diane Sika-Pautonu and I'm the Deputy Vice-Chancellor Pacifica here at the University, and it is indeed an honour to welcome each and every one of you this evening and also to join together in celebration and also an acknowledgement of Professor John Ashton's wonderful achievement and promotion to Professor. Acknowledging also mana whenua, all of our distinguished guests and special visitors who are present here this evening, and we have many, many people who are joining us online this evening. We have loved ones, we have family members, we also have friends, whānau, ainga, kainga, colleagues who are here today in person and who are also linked in online. It's great to have you all joining in this evening. Acknowledging down the front, we have our academic party this evening. We have Professor Julia Horsfield, who is the Dean of the School of Biomedical Sciences. Tēnā koe, Julia. We also have Professor John Ashton himself, who is now a Professor in the Department of Pharmacology and Toxicology. Tēnā koe. We have Professor Paul Smith joining us as well, also from the same Department of Pharmacology and Toxicology. Tēnā koe. We have Professor Ivan Samet as well from the Department of Pharmacology and Toxicology, tēnā koe. Last but certainly not least, we have our Pro-Vice-Chancellor from the Division of Health Sciences, Associate Professor Megan Gibbons, tēnā koe, welcome. So as you can see, there are quite a few people down here at the front, and we have all been tasked with looking after John this evening. And so just to assure you that he is in very, very good hands as well. So thank you for allowing us to share in this evening as colleagues along with him. There are some other very special mentions to make this evening, and it is our privilege to also welcome John's wife this evening, Louise Ashton, who is joining us down here at the front. Welcome, Louise. John's grown-up children, William and Neve, welcome. Thank you for being here. John's sister, who is linking in all the way from Wellington, Karen Connell and her family. Wonderful to have you joining us this evening, Karen and whānau. We also have John's friend, Stephen Chapman, all the way from Oxford in England. Welcome. We have John's other friend, Damon Boswell from Upper Hutt in Wellington, alongside many other friends here and online, and John's colleagues from the University of Auckland. We have Professor Brian Boyd linked in and Professor Mark McKeague. Last but certainly not least, John's PhD graduate, Abigail Bland, welcome to you as well. It's an honour to have you all here this evening as we celebrate together this evening. Acknowledging also the significance of this special occasion for you, Professor John Ashton. And as many of you will know, academia is certainly not for the faint-hearted. And so to have reached professorial status is a wonderful testament, John, of your endurance, your perseverance and your courage. Excelling across domain areas such as research, teaching and service, we are so much looking forward to hearing more about your achievements this evening. As we've come here together to share and celebrate John's remarkable career and journey, we are again reminded that none of us get anywhere in life alone or by ourselves. And so I would ask that you would please join me again in acknowledging not only John, but also his family who have journeyed alongside him thus far. It is now my privilege to hand over and welcome Professor Julia Horsfield, who is the Dean for the School of Biomedical Sciences, to offer further details of Professor John's career before we get to hear from John himself. Malo, thank you for your time.
Ngā mihi nui ki a koe, Diane. Tēnā koutou katoa. Nau mai, haere mai, ki te kaupapa ko te wā. Ko Julia Horsfield tōku ingoa, ko o te tumuaki o te kura biomedical sciences. Biomedical empathy, yeah. Nō reira, nau mai, haere mai. Welcome along, everybody. This is a wonderful privilege for me to introduce Professor John Ashton's inaugural professorial lecture today. And it's an incredible achievement of which we're all very proud. So Professor John Ashton is an outstanding academic in the Department of Pharmacology and Toxicology, and his research career has actually spanned more than 2 decades, and his contributions have made a real impact across pharmacology, neurophysiology, and oncology. Professor Ashton completed his undergraduate qualifications at Victoria University of Wellington, good choice, where he gained a Bachelor of Science with a first-class honours degree. He taught at Waikato University and the Eastern Institute of Technology, and he specialised in the environmental science and biology. And this must have been a fairly interesting diversion for John. He was just saying to me earlier how he was a bit like Better Call Saul of the teachers of Waikato, in that he could stand up in front of any class and tell them about any kaupapa in science that they wanted to hear. So nevertheless, it wasn't John's calling to do teaching in biological sciences, and he then embarked on a PhD studies at Massey and completed his PhD in the year 2000. And then in the early 2000s, transitioned into pharmacology. He moved to the University of Otago, Department of Pharmacology and Toxicology, to take up a postdoctoral fellowship with Professor Smith. The Otago environment proved to be an ideal place for John to succeed, clearly, and he took on a number of research and teaching roles at Otago, published several excellent first author peer-reviewed journal articles while doing so, together with his colleagues at Otago. So no doubt it was this early success in Otago that resulted in John's appointment as a permanent tenure-track lecturer in 2008. But his career in 2008 was really just beginning, and the meeting of the life challenges and the challenges in the lab had only just started. So in 2014, Professor Ashton was diagnosed with ALK-rearranged lung cancer and began treatment with the first generation novel ALK inhibitors. But a remarkable feature of having a career in the biomedical sciences isn't just that a person can really gain the tools and knowledge to understand and also design treatments for not just themselves, but for others experiencing the same challenges. And so because of the substantial knowledge and skills that John had gained, he could make a real difference to his own future and the future of others, both personally and professionally. So you could say that this experience with lung cancer shaped Professor Ashton's future research trajectory. He fearlessly battled cancer on all fronts, personally, intellectually, at home and in the lab. Together with his collaborators and PhD students, Professor Ashton's research has been focused on innovative treatments for ALK-positive lung cancer. The team have been using ALK-positive cancer cell lines to conduct viability and combination drug studies on inhibitors for agents such as IGFR1, MEK, SHP2 and SARC, as well as chemotherapy agents like Pemetrexed in vivo and in vitro. So because of this complicated research, Professor Ashton's leadership and research has been highly collaborative, and this is exemplified by the depth of his international collaborations, from Israel to France, India to the United States. He has led or contributed to more than 90 research grants, gaining nearly $1.2 million as principal investigator. His prolific research output includes over 90 peer-reviewed publications, which are widely cited and respected. Professor Ashton, though, is not just a world-class researcher, he's also a passionate educator. Over the years, he's coordinated and taught across a large range of undergraduate and postgraduate courses, developed novel teaching materials and laboratories, and played a central role in curriculum development. His innovations, like the Drugs and Society course and integration of quantitative reasoning into pharmacology education, demonstrate his commitment to preparing students not just for exams, but for their futures in biomedical science. Professor Ashton has been the primary supervisor of numerous honours, masters and PhD students who have gone on to win national and international awards. Beyond the classroom and the lab, Professor Ashton has engaged with the public on issues like cannabis policy, lung cancer treatment access, and the ethics of drug development. His work has reached audiences not only through academic channels, but also by media, public forums, and government briefings. Despite further cancer battles, Professor Ashton has continued publishing and mentoring, a testimony to his strength and determination in academia and life. His recent focus includes continuing cutting-edge experimental evaluation of next-generation ALK inhibitors, and this work exemplifies translational pharmacology with skin in the game, John's words. Blending personal experience with rigorous exploratory science. Please join me in welcoming Professor John Ashton.
Nā reira, tēnā koutou, tēnā tātou katoa. So I'm going to jump straight into my story with, come on. What? That's what I did. With my beginning of my life, which is in Upper Hutt, which is basically a satellite town of Wellington, and basically a working class, what used to be called working class family and working class upbringing. And that time was a very special time to be growing up in New Zealand, because in the 70s, you could basically run free range, and it was idyllic. And I think that was formative. The other thing that was formative was books, and the first book that really got me really hooked on science was this one by Cosmos by Carl Sagan. Anyway, from there, went to work for a couple of years in Wellington in clerical jobs, and then to Victoria University of Wellington. And I just want to pause here and say, how was it that I was able to do that from my background? Well, it was because in those days, in New Zealand at least, university education was considered a public good. So there were nominal fees, if any. And yeah, that's why. If that hadn't been the case, I wouldn't be here giving this talk today. It wasn't in pharmacology. It wasn't anything to do with biomedical sciences. It was in zoology. That's just a few references to my honours project. And it's where I met Louise, who you've been introduced to. And as I was putting this talk together, I noticed how many of the photos with Louise had her hugging a sort of large furry animal. And as I carried on, I found that the trend continued. And in fact, animals were going to dominate my life, and I'd be competing my science with animals from there. Anyway, we moved to Waikato, Hamilton, where I took up a job at Waikato University as basically a tutor, a teaching fellow equivalent for a couple of years. And I just want to bring in this, because this is really a special part of a sort of time coming towards my work in academia. This, if anyone knows North Island geography, They'll recognise it as Lake Taupo, the volcanic plateau. This is Poriora Forest, and a small, well, it's not that small when you get there, forest called Mapara Forest. And why were we there? Anyone know what that is? That is the North Island kokako. So we were there, Louisa got the job at Department of Conservation over summer, and I tagged along as a volunteer. And there was about 9-10 breeding pairs here at the time, and our job was to basically track to nest and trap around the nest and stop the possums getting into the nest, because it had just been discovered that they eat baby kokako. And in fact, I just checked, and the numbers of breeding pairs there now are up to about 155, which is incredible. Anyway, from there it was to Hawke's Bay, as Julia said, and teaching. I can't even. A little side swipe at some comments from academic colleagues who talk about teaching loads. Well, you try turning up at an institution and being told that afternoon you had to teach a class that you'd never taught before, and there was no teaching materials. You learn pretty quickly. Anyway, that's where Louise and I got married. And then I read another book, which, there was a few in between. And this one inspired me to get back on the science path. And I did a PhD at Massey University, through Massey University. And I haven't got time to explain all this, but there's two things I want to note about it. I did it because I could get a PhD scholarship. So unlike the increasing numbers of kokako, I was fortunate to be there maybe at peak PhD scholarship. So again, if they hadn't been awarded there, I would not be here speaking to you today. The other real highlight is I was looking for a picture of the welcome swallow, which is my sort of experimental test subject. And I found this on New Zealand Birds Online, which is New Zealand Ornithological Society. And I noticed that the reference down here is a paper from my PhD. So whatever happened after that, in my mind, I'd made it to get onto the front of New Zealand Birds Online. But anyway, from there, it's too complicated. Paul knows the story, you can ask him. Basically, found myself basically talked into coming down here, reassured into coming down here by Cynthia Darlington, Paul's wife, who couldn't be here today. And I had other sort of irons in the fire, other job offers continuing on in ecology, but for some reason I thought this might be sort of a better long-term option. I don't know why, but it was a real big sidestep. But I came down we came down in like late 2002, and basically got introduced to laboratory research and found, to my amazement, that it's actually a lot easier than field research because it doesn't rain. You basically, you can organise everything just precisely. Bird doesn't just fly away when you don't want it to. And so in Paul's research program at the time, which was really focused on on the vestibular. That's the inner ear, at least the parts of the brain connected to the inner ear for balance and so forth. Did a number of experiments there and published with the help of Ivan, who was, and the late Ian Appleton, really learnt a lot of laboratory techniques. And then the wheels came off in 2003, what's called a non-STEMI heart attack, which is the one that doesn't kill you but leaves a scar. And then I worked for a while at what's called the Spinal Cord Trust. Learned some cell culture there, and bumped into the now Professor John Reynolds, who gave me some advice on where I might want to be working in the future. So I went back to pharmacology and I spoke to George Lees, who was the head of department at that time, and he got me a job as a teaching fellow at the department. But the important thing here was that, from my point of view, was that he gave me essentially the run of the department, and not the run of the department, but access to the laboratory facilities to carry on sort of doing experiments and in the gaps of my teaching load. At that time, Paul and Cynthia introduced me to Michelle Glass, who's standing right in front of me, and she wrote a grant together, a Marsden grant, which was successful, and that was focused on what's called the CB2 receptor. That's a cannabinoid CB2. That CB is cannabinoid. The CB1 receptor is the one that everyone sort of goes after. It's the one that makes you high. The CB2 receptor is more sort of localized in immune cells. Anyway, this Michelle kind of stepped back quite a bit from this because it wasn't a fully funded, and I was able to really take that forward over the next number of years with help of some very, very talented PhD students. Phil, are you listening? And no, he's not here, is he? Right there. And Jack Alty and Simran Mago. And this was, I remember, I'm keeping it very brief what I'm showing you here. I'm not even going to talk you through it because I know this is a public lecture and I'll say no more about what these experiments often involved. But anyway, it expanded out into many different directions: neuropathic pain, brain injury, and ultimately trying to validate the probes, the antibody probes that we use for this particular thing, following for this protein, basically following in the footsteps of Natasha Grimsey in Michelle's laboratory in Auckland at the time, who had demonstrated that a lot of the CB1 probes, the antibodies, were not valid. And that's basically where this kind of ended. And I just want to reference Paul again here because I remember speaking to him earlier and he said, he gave this analogy, and toss this one to you, Jonathan, of research sort of trends coming in like a wave. And if you catch it as it's dribbling into the beach, well, you've missed the party. But if you catch it as it's sort of cresting, you can really surge into shore and pick up a lot of momentum. And this is really what happened here. And we were really pumping out the experiments and the papers at this point. And 2012 was really a high point. And then all 4 wheels came off. This is, if you know chest x-rays, this isn't a pretty one. That's all cancer, right? So what do you do if you are diagnosed with a nasty disease that gives you a short prognosis? Well, the answer to me was obvious. You collect data. So what this shows is, and there's a reason why I'm showing you this, which I'll get to in a moment, and this is that I tracked my exercise tolerance over time during 2 different regimens of chemotherapy. And you can see here there's a big lot of scatter, but the one thing's pretty clear, they're not going up. Things aren't getting better. Right. 2014, Michelle comes back into the story, actually, because Louise had pushed and cajoled me into emailing, clinical trials to try to find some sort of option, and had heard of this new biomarker called ALK, and I didn't know anyone who researched it, and I emailed all my colleagues, and Michelle referred me to Mark McKeag, who you talked about, and he is an oncologist, pharmacologist in Auckland, and I actually already knew him because I had met him at Auckland department, but also I had examined some of his PhD students. Fortunately, I passed them. But anyway, he was just happened to be studying this particular condition, and he got my biopsy tissue and had it assessed, and it returned a positive result. And then crizotinib, which had only just been developed, sort of a Hail Mary pass here, and I got onto that, and this is what happens with exercise tolerance on a targeted therapy. That's basically over 2 months, and this is how long I can jog until I have to stop. And you can see here it just goes up, and then till everything's okay. And what's going on there is essentially this clearance of the lungs. If you don't know oncological research, you don't know non-small cell lung cancer, this is kind of a minor miracle. This is a penicillin moment. I just was fortunate enough to be living it. So what is ALK? What is ALK? It's related to the receptor for insulin. And what happens in a certain types of cancer, what it's driven by, it's not just a slow accumulation of mutations like you get with the typical sort of smoker profile. It's a sudden inversion of a chromosome. It produces a new fusion gene, which there's a number of different types of these. ALK is just one. And that basically turns that gene on into overdrive and starts pumping out growth and survival signals for the cell it's in. And so it becomes a cancer cell. The flip side of that is because it's such a big on switch, you've got a big off switch if you can block it with a drug, and that's what crizotinib was doing. And you can just see here the timing of this was only about a year before I started presenting with symptoms that crizotinib was FDA approved, which was pretty good. Anyway, so what was the clinical trial results showing? Well, this is the way to read these graphs. These are called Kaplan-Meier plots. You've got time along the bottom here. You've got normalised to about 100% of the population. That is a number of people who are put onto a drug trial. And then each time it ticks down, that's not death, that is the cancer started growing again. And basically, if you compare the 50% point of one to another, so the flatter it is, the better. The steeper it is, it's literally going off a cliff. This is crizotinib, and this is the last chemotherapy I was on. This is best practice chemotherapy now. What I was looking at was this flattening out bit here, thinking that'll do me. But anyway, I wanted to hold here because I was in a more or remission and in a bit of a quandary. I mean, what do I do now? Do I just keep treading water, or do I get back into it? And again, Paul gave some advice, and because I remember you rang me up, Paul, when I was on sick leave or RSL, and talked about another colleague of his in a similar situation who just kept working. And so I just kept writing papers, and I kept thinking. And to be honest, I think some of my highest impact papers of my career were written in that time. I think there was about six papers a year during that time. Paul's absolutely right. It was the best thing to do, just keep moving. And how did I get back into the lab? Well, in 2012, just a year before the wheels came off, I'd read this paper. And I was always trying to shoehorn anything pharmacological back into evolutionary biology. And so when I saw this paper, I was very interested. And I still reference this all the time. I think it's a fantastic study. Basically, it looks at a tumour population as a population of dividing and mutating cells, and that's what drives, ultimately, at least, drug resistance. The idea here was if you could target 2 or more cancer targets, like ALK and something else, that would, if they were to mutate in some way that would cause drug resistance, it wouldn't necessarily cause the other resistance at the other protein. So that was rationale for getting into combination treatments. And what happened in 2015 was, she should be listening, she said she was me, Hayley, who's Hayley Nehoff, who's now Doctor. She's working in the laboratory of Khalid Gresh, who used to be in the department, and she was doing a PhD on glioblastoma and had a lot of kinase inhibitors, which is what crizotinib is in that class. And I emailed Gerard Engelman, who is kind of the grandfather of ALK research, and asked if they could send us their cancer cell line for doing some experiments. And I didn't hear back, but they just turned up a couple of weeks ago. So thanks, Gerald. And so that grew into, it just took off again, a bit like the CB2 stuff, only more so. It was just by the sheerest bad luck, good luck, I was in there right at the start of the sort of build-up of the ALK research sort of wave. And I'll show you one particular example. This is so, going into the weeds is a little bit silly here, but this is just an example. This is Maddie Berry, who's my latest PhD student. If you're watching this, Maddie, you'll remember this, probably, hopefully fondly, but using pretty fundamental techniques, which is basically cell viability assays, if you know this field, and what's Western blot, I was able to get a lot of mileage out of a couple of cell lines. We've got another cell line here, which is because there's two variants. There's several variants of ALK mutations. The first one, the cell line we'd already worked on. The third one, which is a problematic one to treat, we had to buy, which is unfortunate. I just draw your attention to something that's, unless you're an expert, a specialist, I should say, you won't notice. But back in that CB2 era, we're mucking around with antibodies and this Western blot technique. It's basically electrophoresis, transfer the proteins, then you probe with an antibody. If you recall back maybe to that slide, they're a real mess. When it came to the signaling pathways and the proteins involved with ALK, the signal is just crystal clear, and I think it's partly the type of proteins involved, but it's also because they are medical grade. They come sort of pre-screened and pre-tested, which has been really helpful. So in 2019, wheels stay on, but steering and brakes fail. This was when you were giving me a lot of lifts to work, Nels. Crizotinib is a great drug, but it doesn't stay in the brain. It's not a blood-brain barrier penetrating drug. So at that stage, I switched to a drug called alectinib, and this is where Abby, I know she's watching. I have to reference her, I have to say thank you, because she was a complete powerhouse in keeping laboratory productivity going and supervising honours students in my Zoom presence, but not my actual presence for a year, or not a year, but for some time. But anyway, this wasn't an abandoning of a laboratory program and scuttling completely and then starting up again. I sort of managed to keep things going. So what is alectinib? Alectinib is a second generation drug in the same class as crizotinib. It's an ALK inhibitor. And this is one of those Kaplan-Meier plots again. And this is crizotinib down here, and this is alectinib outperforming it in every way. And if you were to put chemotherapy, it would be like this. If you get no treatment, it would be like that. This is, I lost track of my time, but what's notable about this is that another sort of thing I was doing through the 210s was working with a charity called Lung Foundation New Zealand. In fact, I gave the inaugural sort of address in their launch in 2015, and had really pushed the lobby case for funding of these drugs. And it came through in 2019, which was just in time for us. 2021, more trouble, but we just kept going. And then from 2019, what have we been doing? Well, developing our cell culture platforms for drug studies to become more realistic, so to speak, using steroids, which is basically a 3D culture rather than a 2D culture of cells. Bill has been, from chemistry, has been supplying us with various compounds to study. Basically, Bill creates some magical sort of structures and very clever, and we send back the results to him saying that they don't work. But we're getting closer. Anyway, so I want to, at this point, talk about this work we're doing with on the third variant of the ALK gene. We've got a particular cell line which has that, and it's more problematic. People who are treated for this disease don't do as well as the other variants. And this is very relevant because I met this last year another patient who had asked to meet me. He's from the UK, Jeff Otterlink, and his daughter had done our summer school class, and he had got the same disease, and his daughter emailed me and asked if he could contact me and see me. And anyways, we spoke for a long time, and the end result of it was that he wanted to make a donation to the lab. So he's done that. I'm very grateful, very generous of him, and so felt honour bound to try to target the research towards benefiting him directly. I'm not on alectinib anymore. I'm on this drug, lorlatinib. It's a third generation drug, and this is what crizotinib looks like. That's the first one I was on. Chemotherapy would be down here. Lorlatinib is a spectacularly successful drug. But there's a fourth generation drug came out, nelaticib, which is successful. This is called a waterfall plot. Each of these lines, bars, is an individual, and as it's going down, it basically means that their tumour has shrunk by a certain percentage. And what's notable about this is this early clinical trial, all of these people have already failed treatment on lorlatinib. So it's an incredible drug. And I'm waiting for it to, Pharmac to find a reason not to fund it here. But wait, there's more. So lorlatinib can have psychoactive effects. I'm not going to go into details there, but I want to leave this talk with a bit of a high note. Not only in terms of the outlook for this particular disease, but in terms of just how cool pharmacology can get at its best. This isn't work. This is vast teams doing this work, but this structure here, that's lorlatinib, specially engineered to not get pumped out of the brain very quickly. This is the new one, nelaticib. Now, it's been discovered in the last few years that lorlatinib has its side effects mostly through binding also to another receptor called the TRK receptors. And it binds to them almost as avidly as it does to ALK itself. And therein lies the problem. So what the medicinal chemists behind nalaticib have done, they've kept a lot of the features of lorlatinib, but they've added this little witsit here, little ethyl chain, and that essentially there's between these two receptors, the goodie and the baddie, there is one amino acid difference, just one amino acid. And that little ethyl chain, deliberately engineered into the compound, it repels against the amino acid tyrosine in the TRK receptors, the baddies, but doesn't for the leucine amino acid in the same spot in the ALK receptor. So we're down here this is sub-nanotechnology, this is angstrom technology. And so I just think this is one of the most remarkable feats of science, but you won't see it mentioned probably in the news unless you have a direct interest. So this is a timeline of the ALK inhibitor development. And this is about where I came in, and this is about now. And so the discovery was right there, and all these different ALK inhibitors right up to the current day. And I felt like this has been I used a surfing analogy before, I'll use a different one now. I feel like there's been this huge Mack truck barreling along the road, and I've been on a skateboard beside it, sort of being towed along, and managed to get some mileage out of it. So I just want to stop there, and there's too many thank yous to say individually. That's a family about 8 or 9 years ago, at a happy place in Naseby, and our late beloved Labrador Darcy, and that's my mum. At her last birthday, she passed away at 95 earlier this year. So thank you.
Okay. Well, my wife suggested that I ad-lib this, so I hope I don't make a hash of it. Otherwise, I'll have to go home and blame her. My first meeting with John, Professor John Ashton, was back in about 2001. And so I've known him for almost 25 years. And as was mentioned, he worked with us as a postdoctoral fellow on an HRC grant for about 3 or 4 years. And you know, one of the interesting things is that he was actually interviewed over the phone by my wife, who was Cynthia Darlington, who was an associate professor in the department at the time. And despite the fact that he had a background in zoology and no background in anything to do with vestibular or auditory neuroscience, she was convinced that, you know, we had to have him, even though he was at least considering another offer. She came to me and said, he's the one, we've got to have him. And it was because she picked up on his unusual intellectual character. And so anyway, he came down and I connected with him almost instantly from my viewpoint, partly because he had a very theoretical orientation towards things. He would read everything first, and he wasn't really into experimenting for the sake of experimenting, twiddling knobs and working amplifiers. He was interested in theoretical questions and developing experimental methods to answer those questions, very much hypothesis-driven research. And, you know, I was struck by his deep intellectual curiosity, and also his ability to think laterally on the turn of a dime, which really appealed to me. He seemed to be able to go off in all kinds of directions, which I think he's proven definitively now. But at the time, he lured me into cannabinoid research, which I'd never investigated before. But he was interested in cannabinoids and cannabinoid receptors in relation to vestibular and auditory function. And in fact, one of his most highly cited papers, it's been cited 313 times, according to Google Scholar, was on cannabinoids. And then when he left our group, in fact, we continued cannabinoid research on the auditory and vestibular systems for some time. So his interest in cannabinoids kind of rubbed off on us, and so that left a lasting impact. One of the amazing things about John is his ability to pivot and change directions. And in terms of lateral thinking, I don't think you could get a better example than what happened after his health struggles. Because following those health struggles and during them, he made a 180-degree turn to cancer research, which, you know, kind of astounded me. And I mean, obviously, there are all kinds of personal issues involved as well, but I've never seen anyone basically pivot from one area to another, particularly an area as different as cancer research, in such a short time and do it successfully. You know, since then he's published in high-ranking journals like Biochemical Pharmacology. I think it's hard for me to gauge how much it takes to understand at an advanced level a completely new area and then develop a research program in it that finishes up being successful. And so, you know, and he has done all of this at the same time as dealing with serious medical issues that I've said to him dozens of times that, you know, many other people would have been defeated, but, you know, not John. So it's a privilege and an honour for me to say a few words after his IPL, and all I can say is I feel proud and honoured to have him as a friend and as a colleague. Congratulations.
Tēnā koutou, ko Ivan Samet, tako ingoa. Good evening. I'm Ivan Samet. I'm here in what I consider a pleasure in my capacity as head of the department to thank John. Look, I won't say too much. Paul's already stolen some of my thunder, so I'll keep it briefer. But I would like to say, John, first of all, that was an excellent presentation. Thank you. Paul's used the word pivot. I won't say it again. But you have shown remarkable resilience and your ability to deliver new research with excellent findings in a drug translation point of view is commendable. So thank you very much. I'd also like to say I thought I was quite familiar with some of your work. You and I have shared a few drinks and coffees as well. Coffees mainly, but yes. And I've seen you light up when we discussed research, and I've really enjoyed that. So your presentation showcased a number of findings that you had. I think you should also be quite proud of the fact that not only have you delivered important research from a cancer pharmacology point of view, but you've also acted as an ambassador for lung cancer patients, and that's been really valuable. So let me turn around and actually say that I'd like to provide my thanks to Louise for keeping John alive in all of this, for being in touch with us. Louise, you've done a fantastic job, and the credit should go to you as well in all of this. Behind every professor, right? Never mind that, you're important in this. So I'm grateful to you, and also to the boys as well, Neve and to William. Thank you. Gosh, you've changed. You've grown. I'd also like to say that your former students have been a big part. A big part of your life, and you and theirs. And on behalf of Abby and Maddie and all the others that you supervised, thank you. So I'm going to stop here and say, finally, just looking for the thing, congratulations on your well-deserved promotion. And to finish off, on behalf of the university, I'd like to give you this gift as a small token of their appreciation of your work. Thank you, John. Thank you.
There we go. Kia ora, John. Thank you. It's ko Megan Gibbons tōku ingoa. I am the Pro Vice Chancellor of Health Sciences, and it's my absolute pleasure tonight to invite everyone to the Staff Club to join us at the end of this. But I would like to thank you for advising to everyone how hard it is to teach at a polytechnic, but also to take us on that journey from a hedgehog to a receptor. I think you've changed and grown and done what is important to you, and I think that passion came through really clearly in your presentation tonight of you do what's important and you see the importance in it. So thank you very much, and please do all join us at the Staff Club afterwards.